Okay, hello everyone and welcome to the fifth of this year's webinars, today on the topic of chemical proteomics. We're joined by two new speakers, Dr Ilaria Piazza and Patrick Zanon, alongside our guest chair for today, Dr Marcus Bantscheff. Our session today is sponsored by MS Will and we'll be hearing from them before handing over to our speakers. Marcus will then chair the Q&A session after both talks and a roundtable discussion with the speakers who will be joined by expert Stephen Hacker, Patrick's PhD supervisor. So now I just have a few of the usual housekeeping points to go over before we can hand over to our sponsors and then our guest chair to introduce the first speaker. So as always, we're using our Slack channel for questions and discussion. So please join us there to ask questions and use the thumbs up to let us know which questions you'd like to hear answered. Please direct your questions to each speaker by naming them, as we'll have a joint Q&A and roundtable discussion after both talks. For those needing an attendance certificate for this webinar, details will be available on how to get this after the last slide. So once again, we'd like to say a big thank you to the European Proteomics Association, the British Society for Proteome Research, the Young Proteomics Investigators Club, and the London Proteomics Discussion Group Committee for their help and support in setting up this webinar. Thanks also to the London Biological Mass Spectrometry Discussion Group, the London Metabolomics Network, and the News in Proteomics Research blog for promoting this event. And we're also grateful for Imperial College London for providing us with the webinar support. Thanks also to our YouTube channel subscribers. Patrick's talk today will be available to watch again online. So huge thank you to the sponsors, speakers and our guest chair and Stephen for their time today. So now I'll hand over to Eric Verschuren from MS Will, who are sponsoring today's session. So it's over to you, Eric, if you'd like to take control.
Okay, thank you very much. Thank you, Joe. Thank you for the opportunity to show a few slides at least on this meeting. So let me see if this works now when I'm trying to do this live. That's interesting. Did I get control? We're somewhere in the middle of the slide deck, I feel. This is not my slides. So now you've seen them all. We can go start again. All right. So actually, the interesting thing is that before we went through this, hey, getting there? Yeah, now we're there. So before we started the meeting, we were talking and already got the question, what's happening with this? Where can we get emitters? And that's what I focused my talk on today. As most of you know, we're a supplier for mass spec supplies and focusing on proteomics, and we do a lot in the space of emitters, small column ovens, columns, those type of things. So in the interest of time, I'll be quick, and I hope that my slide moves. There you go. So as we all know, the objective has been not forthcoming sending emitters our way the last year or so. And as at MS Will, as we see that we feel responsibility to find new emitters for you, we started working with a company called Covalent. We did this actually already in the fall of 2019 for their columns, and they started making emitters for us. So basically, we've been sort of supplying Europe with 2010 emitters, pooled emitters. And 2 weeks ago, they came to me and said, okay, we've got this self-pack pulled and fritted column now, which actually replaces the picofrit column and is a column that you can pack yourself with a frit inside. So it's really good. On top of that, they've got a really nice line of pre-packed columns, which we also supply with Reprosil in a wide range, starting at 10 micron ID for the single cell people, all the way up to microflow applications. So please have a look at our catalog on the website to see which versions are there. Then switching to the next slide, or at least trying to do so. There we go. And then there's the other format of emitters, which are the tapered emitters. So they're basically the tubes that have a point made to them in different ways. They are supplied either from Fossil Iontech as one of our suppliers here. We also started working with Ryan Kelly, who was a speaker in one of the previous seminars on his micromics stinger emitters, which is, you can see, has an extremely long tip, which is really good for the very, very low flow applications when you're in the very microflow areas. And then for cap flow, we're working with Neomics. And I would like to, why is this so slow? Somehow it's, there you go. Last slide, sample prep solutions, something that's also important. We started working with a company called Proteoform Scientific from Canada, who make a very nice protein purification kit, basically to get rid of things like SDHs and other detergents. And with what are they called? I know I forgot their name. Company in France who make stable isotope labeled proteins, biomarkers, but also a very nice QC protein. And that's all I have to say with my final slide showing some of our suppliers. So there's a wide range of things that you can find on our website. And with that, I'm going to give control back to.
Great.
I think it's Dan.
Thanks,
yeah. Okay. Thank you for your time.
Thanks for that. I'm sure there'll be a lot of people really pleased to hear the news that they can get their emitters from you guys. So, but now I'm going to introduce Marcus Bantscheff, who is our guest chair for today, before he goes on to introduce our speakers. So Marcus Bantscheff graduated in chemistry at the University of Constance and then obtained his PhD from the University of Rostock. Since 2002, he's been at Cellzome, where he led the proteomics team and joined GSK upon Cellzome's acquisition in 2012, where he currently serves as the Senior Scientific Director and Senior Fellow for GSK, leading the proteomics and metabolomics platforms within Functional Genomics Department. His work focuses on the development and application of proteomics and chemical biology approaches to characterize targets, disease mechanisms, and mechanism of action of bioactive molecules. He's an inventor of several patents, has co-authored more than 80 publications, and serves as a reviewer for reputed journals and funding bodies. His research awards include the Life Science Prize of the German Society for Mass Spectrometry and the MCP Lectureship. So I'll hand over to you, Marcus, to introduce the first of today's speakers.
Thank you, Joanna. Hi, everybody. It's a pleasure and an honor to be able to be the guest chair today. And I was asked to give a few words by means of introduction to chemical proteomics. I'll keep it really short to really give time to the speakers that are a more interesting aspect of today's meeting. So chemoproteomics or chemical proteomics is quite a multifaceted subdiscipline of chemical biology. And there's like two main streams, if you like. On the one hand, you have researchers that focus on designing very sophisticated tools to study protein function and pathway modulation. And prominent names that are coming to mind are Caroline Bertozzi and Ed Tate. On the other hand, there's applications that aim to characterize the protein targets of bioactive small molecules, such as natural compounds, but also in drug discovery. And so we can use these days chemoproteomics approaches to characterize target engagement and target selectivity by combining enrichment tools with quantitative proteomic readouts. More recently, there have been quite a number of really exciting innovations in that field. And on the one hand, we are seeing more and more approaches that use direct biochemical and biophysical means to study protein function that do not require designing of specific probes. And those include changes in thermal stability of proteins or resistance to proteolysis. And we are really fortunate to have Ilaria Piazza in here today and present some of her work that she has originally started when working with Paola Picotti on LPMS, limited proteolysis mass spectrometry. Another really exciting opportunity for chemoproteomics is actually the combination of reactive small fragments with proteomic readouts in order to characterize potential targets of specifically designed small molecules. Now, initially starting with fleshing out opportunities for liganding molecules that are otherwise not tractable by conventional chemical means, and eventually by modulating their function such that there would be therapeutic opportunities potentially. This is a very exciting and emerging field, pioneered by Ben Cravett, and there have been substantial contributions by Stefan Hacker's lab over the last couple of years, and we are fortunate enough to have Patrick Zanon present today in this meeting. So without any further ado, let's start with introducing Ilaria Piazza. So Dr. Ilaria Piazza is currently at the Max Delbrück Center of Molecular Medicine in Berlin. She started her career with a PhD studies at the EMBL in Heidelberg, very next door from where I work. And she studied regulatory molecular machines on chromatin structure and gene expression during the cell cycle. And then she stumbled into Martin Beck, and when you stumble into Martin Beck, one of the things you are likely to do is cross-linking. So she did that and started that, and then moved later on to the ETH in Zurich to work with Paola Picotti. Here she developed a method that combines limited proteolysis and mass spectrometry to characterize interactions between proteins and metabolites. She coined it LipSmap. And the goal of her research as a group leader is to study how metabolism regulates chromatin structure and its impact on gene expression. So in her presentation today, she will talk about how proteins metabolites handshake, proteomics technologies for studying. Introduce the second speaker of this afternoon. This is Patrick Zanon from the Technical University of Munich. Patrick has obtained his BSc and MSc in chemistry at the TU in Munich, and he's currently pursuing his PhD in the group of Dr. Stefan Hacker. And I would like to mention that Stefan is available today. He's joined us for this afternoon. He will also be joining for the discussion later on. Really looking forward to that. So Patrick is funded by the Liebig PhD Fellowship by the Funds of the Chemischen Industry, and he's developed the ISO DTB ABPP platform. So ISO encoded desthiobiotene activity-based profiling platform, which allows the residue-specific identification of target proteins of covalent ligands. By applying this method to so far underexplored nucleophilic amino acid, he aims to expand the fraction of the protein that is deemed druggable. And his research is motivated by the need to identify lead molecules and lead compounds with new modes of action to combat antibiotic resistance. This presentation is entitled Profiling the Proteome-Wide Selectivity of Diverse Electrophiles, and I'm really looking forward to this presentation. Thank you so much for joining today, and looking forward to your talk, Patrick.
Thank you very much for the kind introduction. I'm delighted to be here, and also, of course, hello from my side and good afternoon from a very sunny Munich today. I'm excited to present to you today our most recent preprint from the Hacker Lab. Let me jump to a slide. Oh, here we go. Okay, so we in our group, we are very much interested in targeted covalent inhibitors and how they allow us to address proteins that are hard to target with non-covalent interactions alone. So while you usually have this equilibrium of your ligand not bound to protein and being bound, the additional covalent interaction will shift this equilibrium further, and especially in the case of an irreversible conjugation. The equivalency can also improve the selectivity of your compound. For example, the ATP binding site is very much conserved over a panel of kinases. However, these two FDA-approved drugs manage to be selective for specific kinases because of their microacceptors, which react with cysteines in the active sites. Even though industry has for a long time shied away from targeted covalent inhibitors due to concerns that the reactivity will inevitably also cause toxicity, this view has changed, and however, it will still remain crucial for, of course, for the development of drugs to carefully evaluate all the on and off targets of your covalent ligand. So how this is usually approached is by adding an alkyne to a covalent ligand, performing click chemistry after incubation with your proteome to attach an affinity handle, perform the enrichment, enzymatic digest, and then analyze the peptides that you get out of that to identify the targets. However, this has two major inherent drawbacks. For one, you will have to modify your covalent ligand without perturbing the biological activity, which can be very difficult to achieve and can also present some very synthetic hurdles there. Secondly, since the peptide that has actually bound with your covalent ligand will remain attached to the solid phase, you will not gain the absolute information on the binding site. So to circumvent these two limitations, we instead employ a competitive residue-specific workflow, which is based on the pioneering work of the Cravat lab. And in our ISO-DTP ABPP workflow, we start off with two identical samples of our protein of interest and treat it with either a covalent ligand or a respective solvent control, followed by incubation with a broadly reactive alkyne probe. And among these probes, IA and STP alkyne are among the most established, and these will react with a large percentage of cysteines and lysines, respectively, proteome-wide. However, in the sample where we treated the proteome with our covalent ligand, some of the sites will already be occupied by the covalent ligand, and thus blocking those sites from reacting with the broadly reactive alkyne probe. In the next step, we perform click chemistry with these isotopically labeled thiobiotin tags. And since now our two samples will be will have different isotopic label, we can combine them now as they are isotopically differentiated. We again perform the enrichment and enzymatic digest, but now we wash away all these unmodified peptides, and instead, in the next step, under mildly acidic conditions, elute our probe-modified peptides from the solid phase. And the MS analysis now will reveal differing ratios of the modified peptides for the heavy and the light samples. And this ratio very much depends on how quantitative the covalent ligand was interacting with our sites, and how much it was succeeding in blocking the interaction with the broadly reactive alkyne probe. So the ratio that we get out of this directly relates to the target occupancy of the covalent ligands. And taken together, this workflow thus allows us to streamline the identification of the exact binding site of our unmodified covalent ligands. This allows us also to pursue studies of structure affinity relationships proteome-wide. And since we now have a workflow in hand where we can actually pinpoint the residues that our probes are binding to, we can also start thinking about whether or what the preferences are for these electrophiles to react with the proteogenic nucleophiles. And this is what we did in our preprint, which is available on ChemArchive. And out of all the proteogenic nucleophiles, cysteine has the most nucleophilic side chain. This means that it is quite easy to come up with, skipping back. It's the most nucleophilic side chain, and accordingly, you can quite easily come up with electrophiles that allow us to address cysteines. And this is also why the development of covalent inhibitors so far is very much focused on cysteines, just because it's a very straightforward thing to do. However, cysteines are quite rare in proteomes, and this means that we inherently target our potential targets based to that subset of proteins which bear a suitable reactive cysteine. So accordingly, if we also take into account all these other nucleophilic side chains that we can address, we would very much expand the number of potential sites we can address with covalent inhibitors. And many groups have worked on this to come up with chemistries that will selectively engage these side chains. However, many of them have only been used or tested with peptides or isolated proteins, and even if they've been used in the context of a proteome, it is very hard to compare different studies amongst each other because of the differences in the experimental workflow, in the instrumentation and the data analysis. So we figured it would be very interesting to have a resource that would allow us to compare all these different probes. And the experimental workflow that we came up with is very much similar to the workflow I just showed you. Here we start with two identical samples of a protein of interest, For us, this is Staphylococcus aureus, simply because we're very much interested in the development of target covalent inhibitors to combat antibiotic resistance. And this will, of course, result in 2 identical modifications in both samples. However, then we introduce our isotopic labels and perform all the other subsequent steps as before. As indicated here, we will now, in a mass spectra, get these double peaks, which of course result from the isotopic labeling of light and heavy with a spacing of 6 Dalton. And these double peaks now very confidently allows us to assign whether a modification that we detect for any peptides actually stems from interaction with our probe, or if that's stemming from some cellular processes or whatever. For the analysis of our data, we started a collaboration with the group of Alexei and Swisky, who developed the Frag computational platform. And the way we do our data analysis is in 3 basic steps. First, we start off with an open search, which is a very unbiased approach. We just look at what the modifications are on the peptides that we detect. And as you can see here, the main modification corresponds to the expected mass shift, which corresponds to the nucleophilic substitution of the iodide here. But you can also see that we detect other modifications, namely the oxidation of the formed thioether, the formulation and carbamidomethylation, which is just modifications that can occur during our workflow. I also want to mention here that if you have an electrophile that you're not really sure how it will react with proteogenic nucleophiles, so you don't actually know what the modification is, this search will provide you with a mass shift value accurate enough so you can actually deduce the chemical formula from that, which is pretty cool. Then in the second step, we perform an offset search, which means that we now limit the search to those peptides that have the mass shift or the offset, which corresponds to the modification that is interesting to us, and then localize that to the respective residues. Here you can see that the modification that occurs here is very selective for cysteines, which was to be expected since uracil alkyne is a very established probe to profile cysteines. In the rest of the talk, I'm going to present you these selectivities in the form of such a ladder plot, where the height of the 1-letter amino acid code corresponds to the percentage of the modified sites. And on top, you also have here the number of total modified sites. So this also includes all the other amino acids that we detect in this search. However, I want to be clear that you don't mistake this number here for the number of sites that we can actually quantify. For quantification, we run a closed search, which now means we not only limit our peptides to those that have the respective modification, but now we also tell the software that we want the modification to that the modification occurs on cysteines. And oops, and you can also see here that depending on how we mix light and heavy, usually we do 1-to-1, and here is a control we also did 4-to-1, and in both cases all the quantified sites we detect nicely distribute around the expected values. So we did this analysis for a total of 54 alkyne probes, and during that we collaborated also with the groups of Dean Toast, Chris Chang, and Katrin Lang. And here is an overview of all the probes that we have tested. And here I really want to emphasize that the underlying chemistries for all these electrophiles have mostly been developed by other groups. And even some of those exact probes that are shown here have been synthesized by other groups. So if you're really interested in a particular electrophile, I want to invite you to check out the references that I've given on the respective slides. So as I said, it is fairly easy to find electrophiles that will react with cysteines. For example, these alpha halocarbonyls, these compounds that undergo nucleophilic aromatic substitutions, these hypervalent iodine compounds, which have been developed by the group of Jerome Waza. And also here are shown unactivated compounds that undergo SN2 reactions. In all these cases, you see there is a preference for cysteines to some varying degrees. You can also very well see that there are huge differences in the reactivity of the respective probes. We have also looked at microacceptors, since they are the current mainstay of the targeted covalent inhibitors. And again, you can see there is a preference for cysteines with some varying selectivities for some of them, and again, very huge differences for the reactivity. For lysines, the most commonly employed agents to selectively monitor them are acylating agents, and we were able to confirm the good selectivity for lysines here. And furthermore, could also show that these ascorbic acid monoamides also are very selective for lysines, with these alkyl scorates being very promising for the development of or to be used as targeted covalent inhibitors, since they combine the high selectivity with the moderate reactivity. When you think about chemistries that can selectively engage amines, you will of course be thinking about the formation of imines. For the use as probes, however, we require a very irreversible reaction to occur. So the modification of occurring by the probe will actually be very much irreversible. So it is very advantageous to have a residue in the ortho position that will lead to the formation of a stable product, as like here. So similar here is this nitrosobenzaldehyde, which is generated through UV irradiation of this probe, leading to a stable and selective modification of lysines. And I also want to stress here that these photoactivatable probes show high promise for the application in live cells, since they are non-toxic until you actually provide that stimulus. So you have spatiotemporal resolution of the activation of your electrophiles. Something I forgot to mention, but to be clear about that, the red circles actually indicate sites of the initial nucleophilic attack. So just to give you a better understanding, also for the probes that I didn't display the mechanism for how they react. Of course, the N terminus also presents an amine that can react with aldehydes, and we were able to show that the stabilization through this amino species here actually allows us to very selectively monitor N termini. However, the number of sites that we detect here is rather low, so this is definitely something that we hope can be optimized in the future. Building on our previous efforts to monitor aspartates and glutamates, to selectively monitor aspartates and glutamates with these light activatable tetrazoles, we now also have developed this hydrazone chloride, which does not require light activation to form this reactive nitrilamine species, which will engage aspartates and glutamates selectively. Interestingly, these azarines, which also selectively react with aspartates and glutamates, show a very different ratio between those two amino acids, which might come from just steric reasons, since obviously aspartate has one CH2 group less, and thus the probe might not get as close to the residue for these probes here. For these probes that undergo substitution at sulfur 6 centers, we were also able to confirm the varying selectivities of reaction with tyrosines and over lysines. And those probes, which have been very much pioneered by the groups of Barry Sharpless and Ken Su, those probes engage the phenolic hydroxyl group of tyrosine. However, there are also chemistries available that interact with the aromatic core of tyrosine, you can see that this modification is very selective for tyrosines. However, when we look at the open search results, we could also detect another modification to occur. And this modification corresponds to the isocyanate being formed, which will then react with lysines and N-termini to form this urea. And this is one of the examples that we also provide in our paper to show that this unbiased open search approach really allows us to catch such events. When we were thinking about the electrophilic aromatic substitutions, we were also, of course, considering diazonium salts, which are most renowned for such chemistries. However, we were not able to detect much of this diazo modification, and instead we found as a main modification to occur the arylation on cysteines and aromatic amino acids. And interestingly enough, we also can see quite a few phenylalanines here, which is something that we didn't expect initially. Yeah, these pyridinium salts upon UV irradiation undergo radical mechanism, and this allows us to very selectively monitor histidines and tryptophans, two amino acids that up to that point were very hard to get a grasp on. And we are very happy to now have a probe in hand to monitor them proteome-wide. And we were also looking at these kind of probes, which upon light irradiation form these orthoquinone methites, which undergo formal 4+2 cyclo addition with tryptophanes and allow us to further profile that residue or that amino acid proteome-wide. The groups of Chris Chang and Dean Toast have developed these oxaziridines for selective binders to methionines. However, when testing this probe, we were unable to detect the respective modification or any modification at all for that matter. So we started collaborating with them, and we came up with this improved probe, which now allows for a more stable modification and allows us to monitor a number of methionines proteome-wide. The last probe that I want to present to you today is this phenylglyoxal probe, which interacts with or engages arginines and produces this very selective modification for arginines. This is also the very first time that we were able to monitor arginines proteome-wide. So taken together, we have a set of probes that allow us to monitor a total of 9 different amino acids and the N-terminus. And since we recognize that not everybody is as interested as we are in bacteria, we also performed the respective experiments in human cell line. And as you can see, the selectivity is very much pulled up also in this proteome, showing that this is a more general thing and not just specific to a particular proteome. The only exception here I definitely want to mention is the probe that allows us to monitor N-termini. So even though I didn't go into detail on the improvements that have been made on the computational platform that is FragPipe, I really want to emphasize that this was very crucial for the project. And I'm very certain that FragPipe as a tool will be very valuable or continue to be very valuable to the field. And we have this set of probes in hand for the selective monitoring of 9 amino acids and the N-terminus. Among those, as I pointed out earlier, we have the very first probes to monitor arginines, histidines, and tryptophanes. And we envision that this set of probes that we now have will be very handy for the development of covalent inhibitors that go beyond the residues that have been really addressed so far in the form of these broadly reactive alkyne probes in a competitive workflow, as I described earlier. And we very much hope that in the future we will have chemistries available that also allow us to selectively monitor serines, threonines, and the C-terminus to further expand on all the residues that we can address and monitor. Finally, if this wasn't enough data for you, I very much invite you to check out the supplementary files online on ChemArchive. I want to close my presentation by thanking everybody who contributed to this work, both in Munich and in the US. I also very much want to thank the organizers for putting this webinar series together and also for inviting me for today's session. And lastly, I want to thank everybody in the audience for your attention. And yeah, I'm very eager to answer any questions you might have. Thank you.
Thank you, Patrick. Thanks for demonstrating how you can combine really exciting chemistry with cutting-edge proteomics to scout through the amino acid portfolio and its reactivities across the proteome. I suggest that we now open the discussions, and there's been a couple of questions coming in for both presenters. I'm wondering, it may make sense to start with a couple of questions to Ilaria first, and then go back to Patrick, if that's okay for everybody. Just trying to browse through what came in lately. So there was one question that was like twice, and I'll just directly start with that one. It's one for Ilaria, I guess. Tate started with it. Is does in-cell chemical cross-linking enhance or reduce the LIP signal for protein complexes? Now, have you considered LIP following, for example, fractionation, IP, or in-cell proximity biotinylation to focus LIP analysis on an interactome or subcellular proteome of interest?
All of these are wonderful ideas. So, we are looking into some of them, but we have the preliminary data to present it now, but for sure there will be developments in the future. Cross-linking, we haven't looked into this specifically, but
right.
There is always this problem of, the tip of the iceberg in proteomics. So if you fractionate, for sure you can increase the sensitivity towards a certain targets or subcellular compartment.
I guess one of the aspects, and I'm not sure if that was keen on that, but it was something that came to mind when I was thinking about it. It was since for LipMS, you always need to first slice the cells and then run proteolysis. So whatever in-cell situation you had, you can't freeze it. But now using intracellular cross-linking at low concentrations to stabilize a specific conformation, then interrogating that conformation with proteolysis could be an exciting opportunity to combine the two things that you spent some of your life with it.
Yeah, there is always a question of finding the soft spot, because exaggerate with the cross-linking, then you might somehow destroy the structure of alter them artificially. But that's also a dilemma that you have when you do cross-linking MS, no? Or any other cross-linking strategy. So it's for sure a way to try to improve things. Thank you.
All right. Joanne, I think Joanna or Anonymous put in a question. Is it possible to study GPCR receptor and drug interaction using LipMS? What are the chances of spontaneous hydrolysis if the binding is reversible with ligand? Yes, it's two questions. You may want to start with number one.
Okay, so maybe one could be generalized to membrane proteins.
Yeah, that was the question I put in, so thank you for taking that up.
So of course, membrane proteins are, the holy grail. Everybody wants to study them well because they are important druggable targets, but they are also very hard to tackle. Especially with these label-free methods, because they all these methods rely somehow in keeping the environment as native as possible, and the membrane compartment is not really accessible, or you cannot really solubilize it easily without perturbing the structure of the membrane protein. So we have had some success with the membrane protein with the target called prosilardin, that is a drug that binds sodium potassium ATPase pump. But in that case, we had to adopt a strategy that one of the attendees suggested before. So first, isolate the membrane compartment. So using this kind of concavalin beads or lecithin base, and then increase the amount of these proteins that we have in the mixture. For what we can detect, we are always limited by the domains or the regions of the proteins that are accessible by the protease. So for sure, the transmembrane domains that are crossing the lipid bilayer, we have a bit of troubles because the protease will not go in. So on the other hand, the extracellular domains are those that are normally involved in interacting with the drugs. So it's always a hard class of target to deal with, and plus plasma membranes are not really abundant in the cell. So I personally think that the only way to tackle the problem of this with this kind of approach is to perform an enrichment of some sort at any point of the protocol pipeline. Or maybe something that you have recently published for TPP, Marcus.
Didn't want to mention that too, obviously, so thank you for doing that.
I really like that work, by the way.
Thank you so much. All right, so what else do we have? There were a little bit of discussions on the dose response and the targets that you found. So when making this into an compound profiling effort, right? I think you really nicely described it, or very adequately described it. It's like you get a dose-dependent change in the lipid peptides. I just wondered if you could share a little bit more how consistent the dose-dependent change is across different peptides you might identify for the same protein, how representative the dose-dependent changes are for affinity, and how you think that characterizes selectivity of inhibitors.
So first of all, there is always a question of coverage. So like for those proteins for which you can detect a decent amount of peptides, then you have also higher chances to have more than one lead peptides. And in that case, it's provided that you have good signal. So also, you have also done the experiment with a sufficient amount of points and with little noise, and the fitting is good. Then in general, this IC50 that you can extrapolate is consistent among the peptide. You can have, a variation of 1 order of magnitude because of the think a bit, and this is always a problem with this kind of fitting strategies. So, and one has to improve the statistics and the way you do that and the model that you use. But it's intrinsic in the approach that you adopt in that sense. And then what was the second part of the question?
Selectivity.
So if you, let's say you have a compound that binds with low selectivity and one that
yeah, it's like how clear can you discriminate? So you got one, you got whatever dose dependent, an inflection point of micromolar, you got another one that is a lot less. How informative is that?
So for example, just to give the most general one that I can think of, if you do this with metabolites, you could have numbers around the millimolar even range, that makes sense because they are typically not binding very strongly, at least some of them. While instead, when you do this with drugs, you could go into the nanomolar range. Whether this, overlaps, completely and precisely with the data that is published that comes with other methods is not always the case. And I think this is a bit of a problem that you have also with other proteomics methods, because they are not really direct binding measurements. There are some somehow indirect. So if you think about what happens here, you have a protease that binds and nick, and then what you are measuring, or in a quantitative way, is the effect of the cleavage of the protease. So what happened typically is that you underestimate somehow the strength of this binding. And I think this is also something you observe with TPP, correct? Or
we observe it even more so in things that are just happening on lysates. So if we do experiments in lysates, being that kind of beads or any of those chemoproteomics competition binding experiments, it's the same story. TPP, probably also.
Yeah, and then, you know, that's also what you mentioned, like, you know, it's not really an in vitro system, super controlled, like, you know, how would you would typically do a binding assay, you know? And so in a lysate, you could have other competitors or other proteins that are competing for the molecule that you're adding, and these are also secondary effects that you cannot really exclude that they are not happening. So in a way, this is a more bona fide measure of what could happen in vivo with you know, all the environment. But then it's also it's hard to judge what's the better representation of the truth.
Yeah, ground truth is difficult to control. So let me just ask you, I suggest we have just one more question directly to Ilaria, then go to Patrick, and then we just take it as for the rest of the session as it comes. So there is one question here. Dear Ilaria, thank you for the great talk. Could you please comment on the sensitivity of LipMS to the abundance of the protein target and its cellular location? Can low abundant non-cytosolic protein targets, okay, a little bit close to what we said before, be investigated? Maybe you want to focus on the abundance in particular and other cellular locations. I think we've covered the membrane.
Yeah, I think we covered this in both papers. So, like, of course, I think even you know, more importantly than with other methods, we rely a lot on having a good peptide coverage. You know, a bit like with phosphoproteomics. No, so the more abundant the proteins are, the more likely you have to measure more, you know, peptides that are mapping in different domains and have a, you know, complete coverage of the whole sequence of the protein of interest. And, you know, the more frequently you can be in that situation, the more likely you are to be able to detect a potential compound binding to the protein and to the domain that specifically binds it. And then we, you know, just to give a rough number, I would say that, you know, when you have more than 20-25% sequence coverage for a, you know, potential target of interest, then you have good chances of detecting something, at least. We kind of estimate the medians that we need. But sometimes, if you're lucky and the peptides are really covering the regions where all the fuzz happen, it could also work with lower sequence coverages. But this is, you know, the stochasticity of the method.
Thanks for that. Just while closing, there is a late coming question about whether or not you've performed actually LipMS on individual proteins and how that compares to top-down MS of a protein drug complex. Also relates to another question was put a bit earlier up that asks to generalize what kind of approach is best used for localizing where a drug interacts with the target.
So yeah, I mean, this goes a bit in pair with the previous question. Of course, one way, an alternative to increase your power is somehow to perform the experiment in a targeted way. So you can, if you can afford to produce recombinantly or isolate from source a complex or a protein of interest, that's definitely a strategy to increase the coverage. So have more molecular details. But of course, then the background you're working with is slightly different. So we have observed that one has to typically change a bit the ratios of substrate to protease and adjust this a bit to avoid over-digestion or under-digestion. And of course, here you are more dependent also on how sturdy is the protein to proteolysis. Of course, if a protein is unstructured, then it will be chopped off very easily. If a protein is instead a single domain one, like a GFP, a stone-like protein, then you have to adopt a different strategy. But then you can afford to test more and have more information.
Excellent. Thank you so much for the answer and for the questions. Thanks very much to all those who've contributed to those. I suggest we change topics ever so slightly and move over to Patrick and Stefan's topic. Maybe I can just give a start with one. I'm really happy you did these experiments, by the way, because it really addresses an element that we discuss so often. Is like, how do the different reactivities pan out? Now, if we focus on cysteine for one moment, because as you said, like everybody loves it in a way. You've used a range of different reagents, and a question to me is like, do you see gradual differences, quantitative differences in the cysteine coverage just because you got more reactive versus less reactive, or do you see true qualitative differences in the covered cysteinome by just changing the reactive group? So is there a fundamental difference between using, you know, a iodoacetamide type of reagent versus an Michael acceptor versus another more elaborate molecule in terms of what you can access in this reactive cysteinome?
I mean, in general, every probe will have some sort of like slight bias. Some binding sites will be more readily accessed with something greasy or something more slink, for example. But in general, especially for cysteines, we tend to get a very good overlap in the sites that we can detect. However, if you really want to cover more ground, then it does make sense to use different probes because of these slight differences, and also for the vast majority of the analysis that we've run, we've analyzed duplicates, which of course you can expand on just to get more replicates in, and there's plenty of work out there that if you increase the number of replicates, or let alone the MS runs, that you can increase those numbers. So if it's really the overall coverage in the proteome that you're going for, then yeah, I would definitely advise doing more replicates and also using more complementary probes. For most probe types or amino acids, we have also these Venn diagrams in the supplementary information, which can be helpful to assess which probes are the most complementary.
Thank you for this. There was another question to Patrick and Stefan. Can you use this Frag pipe analysis workflow to profile photoaffinity labeled proteins? I guess
yes. So we have some projects where we started to do that. In general, the modification that occurs can of course also be detected through his workflow. In general, of course, these photoaffinity labels are much more specific than what we have shown here. We've very much focused on these broadly reactive probes to really be used in a competitive drug discovery approach. But yeah.
There's a cool question I wanted to get at both of you, and this is, I'm interested in the selectivity of some of the covalent probes. I expect there may be instances where a broad spectrum probe may label other residues that are not picked up because the modifications are not stable to the workflow. Have you looked into that?
I mean, it's hard to really look into that because our readout obviously is biased for these modifications that are stable. But obviously this is, some might argue, a limitation to this workflow. But I really believe that for the development of these broadly reactive probes, it doesn't really matter that much, because we still are able to monitor all these lysines, for example, with activated esters. And even though these activated esters might react with cysteines, this doesn't change that we can pick up on the competition that we see on lysines. And yeah, I mean, you can also spin this further for For covalent inhibitors, if you want to do this analysis with really something more inhibitor-like, that of course there are plenty of examples where activated thioester is being formed has physiological relevance. But I would also argue that there are other modifications that can occur but are also very quickly falling off. If you, for example, think about microacceptors, they for sure can also react with serines, but the retro-micro reaction again is very quick. So even though there might be some reactivity to other amino acid residues, it is a bit tricky to figure out whether that is actually of physiological relevance to inhibitors. But still, our competitive workflow really holds up for this. If you want to profile cysteines and the formation of thioesters on cysteines, then you will just use a cysteine specific probe.
Right. So your expectation is that the real drugs would employ slightly different chemistry than what you're using for your probes, as you are aiming to change ultimate sensitivity, selectivity with the reaction.
Yeah, very much so. I mean, for inhibitors, it is without having any idea of the active site, it can be very hard to predict which will be the actual residue that interacts with, because even a residue that in the proteome-wide sense is not very reactive towards the electrophile, we might still, through the very strong non-covalent interactions, position that residue in a way that it will still react with the electrophile.
Mm-hmm. Thank you for that. Here's a question, slightly different topic, quite relevant for many though. Do you find any issues with the reproducibility of the biotin-avidin enrichment pull-down illusion? For example, contamination with avidin peptides, batch-to-batch avidin issues. Which avidin have you found to work best in context of your work?
Yeah, so it has been a while since we compared different beads. I think we tried 3 different commercially ones. We stuck with the high capacity strapped avidin agarose beads by Thermo Fisher. I'm pretty sure that in the supporting information you will also find the cataloging number if you want to order those. And those work the best for us with the let's say carryover of peptide of strapped avidin peptides. I'm actually not sure, because we are specifically looking into modified peptides. So this is not something that will turn up in our usual data analysis. You could, of course, look into all the data again and see if you can actually find it. But in that sense, it doesn't affect us too much in our downstream data analysis. Yeah. Did I miss something from the question? I'm not sure. It was several questions packed into one.
Well, I guess you can discuss for ages about biotin strapped avidin workflows, and I guess many of us have wasted a lot of their time in trying to optimize it. And I guess you've addressed part of that in the biotin that many people are using, at least in many people's experience, as helped addressing a lot of it. So I guess we just leave it on that. I would have a very basic question to you. When you think about targeted covalent inhibitors, the beauty of cysteine is you got a relatively small hyperreactive cysteinome, and a majority of cysteines are kind of silent. They're not very reactive. Either they are engaged in disulfide bridges, or it's just not super nucleophilic and occupied by other means. They're also tightly regulated in their activity, right? You get the sulfines and sulfones, you get the glutathionatics and all of that. Now, it's a little different with other amino acids. So if I think about lysines, they're mostly decorating on the cell surface. And I really wonder, I mean, obviously we also know that catalytic lysin of kinases can be targeted and can be relatively nicely targeted, but what we need there is a really good binding pocket to optimize that around. So when you look at these different amino acids, for which of those amino acids do you think that the difference between the on average reactivity and the maximum reactivity you can achieve in such a binding pocket is the highest? So which of these additional amino acids would be really worth developing into targeted covalent strategies for medicines and for inhibitors? And which of the others, which of those would you think it's really, really hard to get that level of discrimination in because of the reaction or the type of the amino acid?
Yeah, very, very interesting question. I mean, in general, if you think about the side chains that are to be considered more basic, like lysine and arginine, of course, you have a much deeper gradient, if you want to put it like that, in the reactivity depending on what surrounds the residue. So if you have a very surface exposed arginine, for example, then it will most likely not react in any way nucleophilically. It is a little hard to really generalize, because I think that also for arginines, there are viable targets out there that you can address with covalent inhibitors. But I think also because lysines are very, very common, they make up a large percentage of proteins, they will probably make up the after cysteine the next most common group to be targeted. And after that, I can also see, for example, tyrosines to be among the most interesting ones. But yeah, like I said, in general, I think that essentially every amino acid side chain holds some promise for the development of covalent inhibitors.
Thank you for that. I got some 5-minute warnings coming up, so I guess we need to wrap up soon. Stefan, I saw you smiling, so I was just wondering if that smile was indicating that you may want to comment as well.
Yeah, I mean, this is kind of a hard question. What's the best amino acid to target, right? I think we know from a lot of work. I think if you come back to your initial question, where has the largest difference in reactivity, then you probably have to say serines, because serine hydrolases are really uniquely reactive compared to all the other things that are on the surface. But of course, if you look at that catalytic nucleophiles, you're also limiting yourself very much to specific subgroups of proteins. So if we think more about not the ones where there's like a specific nucleophilic reason for them being more reactive, I think then beyond cysteines, really the ones where at physiological pH you don't have the most reactive species are probably the ones that are most promising, like your lysines and your arginines, where most of them will be protonated, but then there will be a small subset that's maybe not protonated or not as protonated as at physiological pH. I think that gives you a unique chance for good selectivities, and I mean we've shown that in the paper back with Ben Cravat still that there is also for lysines a quite small set of like very reactive lysines that you can get to, and then even with activated ester probes, you can very specifically label that lysine versus the lysine to arginine mutant of the same protein. So I think there's good chances, especially for those. But yeah, I mean, there's of course also beautiful work by others for residues that you just wouldn't expect to be targetable selectively. So I think, yeah, as Patrick mentioned, it's probably mostly on a case-to-case basis, but I think those are the ones where you probably, broadly speaking, over the whole proteome have the best chances.
Excellent. Thank you so much for the answer. I guess we need to bring this to a close if I interpret the conversation in the background correctly. So let me just thank again very much to both the speakers, Patrick and Ilaria, also Stefan for joining us for the discussion. Thanks also to all of those of you who've contributed to the chat and making this a really lively conversation and a good discussion. Please also use the opportunity to continue asking questions in Slack. I'm sure there will be an opportunity to quickly look into this. And with that, I think I hand it back to Joe, is it?
Yeah, thanks, Marcus. Yeah, so yeah, please continue asking questions on the Slack. Hopefully our speakers will get there and answer any of the ones that we didn't manage to get through during the discussion. So for those of you who need a certificate of attendance, there will be one just in a few minutes after we wrap up. So yeah, thanks really loads to our speakers, our guest chair Marcus and Stefan for the lively discussion, and to MS Will for sponsoring today's webinar, and Eric for presenting his slides. Thanks to everyone who came along and those committee members who've been working away in the background to make this webinar possible. We're taking a little summer break for July, so we'll be hosting our next webinar in the series in August with a session focused on structural proteomics. So keep an eye on our website and social media for more details once we've